A German scientist named Otto Warburg discovered something about cancer in 1923 that most of the medical world chose to ignore for the next 70 years. What he found was that cancer cells eat sugar differently than every other cell in the human body, consuming glucose at rates up to 200 times higher than normal tissue, even when plenty of oxygen is available. They don't do this because they need to. They do it because it helps them grow. That single observation, made over a century ago, turned out to be one of the most important discoveries in modern cancer biology. It is the reason doctors can locate tumors on a PET scan, and it has led to multiple FDA-approved drugs. More than that, it opened the door to 7 distinct metabolic switches that researchers are now targeting to slow or stop cancer from growing. Today, we are going to walk through all 7 of those switches, what they are, how they work, and which ones already carry FDA approval. Pay special attention to switch number 7. It may be the most elegant solution of all, and the most overlooked. Before we go any further, a quick note. This video is educational, not medical advice. Always consult a licensed professional before making any changes to your health or treatment plan. Nothing here replaces your doctor. What we are doing is exploring verified, published science so you can have better conversations with your care team. With that said, let's get into it. Otto Warburg came into the world in the year 1883 to Freiburg, Germany. He was born into one of the most extraordinary scientific households in Europe. Here is the discovery stripped down to plain English. Every cell in your body runs on fuel, and that fuel is glucose, the sugar that comes from the food you eat. A healthy cell sends glucose through a long, efficient process inside structures called mitochondria, which you can think of as tiny power plants. One molecule of glucose goes in, and roughly 30 to 38 units of energy called ATP come out, clean and efficient. What Warburg discovered in 1923 turned that understanding sideways. Cancer cells skip the power plant. Instead of routing glucose into the mitochondria, they take a shortcut and ferment it, the same way yeast ferments sugar into alcohol. That shortcut yields only 2 units of energy per glucose molecule instead of 38, and it churns out a waste product called lactic acid. In his original experiments, tumor tissue pumped out approximately 70 times more lactic acid than normal liver, kidney, and heart tissue. The part that baffled scientists for decades was that cancer cells did this even when oxygen was present. Normal cells ferment only when starved of oxygen, but cancer cells ferment constantly, choosing the less efficient path on purpose. Think of it this way. Imagine you own a factory, and you have a choice between running one large, efficient machine that produces a lot of product slowly, or running 100 small, fast machines that each produce less but collectively let you expand much faster. Cancer cells pick the 100 small machines every time. The shortcut is wasteful per molecule, but it is fast, and the byproducts become raw building blocks, the nucleotides, amino acids, and lipids that cancer cells need to keep dividing. That is the Warburg Effect. Scientist Ephraim Racker coined the term in 1972, and today, clinical data from PET scans confirms that over 90% of human cancers show increased glucose uptake, consistent with what Warburg first described. Every PET scan performed on a cancer patient, millions each year, works because of what Warburg saw a century ago. The radioactive glucose tracer floods into cancer cells at wildly elevated rates, and the tumors light up on the screen for exactly the reason Warburg identified. If the discovery mattered that much, why did the medical world sideline it for decades? 2 reasons explain the exile. The first is that Warburg pushed his idea further than the evidence allowed. He didn't just say cancer cells metabolize differently. He declared that damaged metabolism was the prime cause of cancer, that when a cell's mitochondria suffered injury, the switch to fermentation was the event that created the disease. Genetic mutations, in his view, were secondary. Modern science has shown this was only partially correct. In most cancers, genetic mutations in oncogenes and tumor suppressors are what drive the metabolic shift, not the other way around, and cancer cells' mitochondria are generally still functional. But, and this part is worth sitting with, there are specific cases where Warburg had it right. Mutations in metabolic enzymes called SDH, FH, and IDH can directly initiate cancer. His idea wasn't wrong. It was incomplete. The second reason came down to timing. Starting in the 1970s, cancer research pivoted hard toward molecular biology, with scientists chasing oncogenes, tumor suppressor genes like p53, and the Philadelphia chromosome. Metabolism got pushed aside, or as one researcher described it, relegated to the backwaters of cancer biology. The rehabilitation started around 2006, and three forces pulled metabolism back into the spotlight. First, the daily clinical use of PET scans was validating Warburg's observation millions of times over. Second, scientists discovered that the most well-known cancer genes, AKT, PI3K, MYC, and HIF, directly control metabolic pathways. And third, researchers in 2009 found that mutant metabolic enzymes produce substances they named oncometabolites, molecules that directly reprogram cells toward cancer. Douglas Hanahan and Robert Weinberg sealed the comeback in 2011 when they added reprogramming of energy metabolism as an emerging hallmark of cancer in their landmark paper. Citation data tells the story in numbers. Warburg's 1927 paper collected fewer than 10 citations per year for decades. By 2021, that number had climbed past 500 citations in a single year. Now, let's talk about the 7 switches. If the Warburg effect describes how cancer cells rewire their metabolism, these 7 switches represent 7 different points where researchers are trying to rewire it back. Think of your body's metabolism as a terrain, an internal environment. Cancer doesn't depend only on DNA mutations. It depends on the metabolic conditions around and inside the cell, and changing those conditions may change the disease's ability to grow. Here are the 7 switches. Switch 1 targets glycolysis directly. Switch 2 activates AMPK through metformin. Switch 3 goes after glutamine metabolism. Switch 4 inhibits the mTOR pathway. Switch 5 blocks HIF-2 alpha. Switch 6 targets IDH mutations. And Switch 7 uses DCA to inhibit PDK. We are going to cover each one. And keep Switch 7 in the back of your mind as we go, because it is the one that most directly reverses the Warburg effect itself. Switch 1. Targeting glycolysis directly, cutting off cancer's favorite fuel. This is the most straightforward approach of the 7. Cancer cells devour glucose at rates 10 to 100 times higher than normal cells, and Switch 1 says block the enzymes they use to process it. A compound called 2-deoxyglucose, or 2-DG, acts as a glucose lookalike that enters the cell but can't be fully processed, jamming the system from the inside. Another compound, 3-bromopyruvate, goes after hexokinase 2, a critical enzyme in glycolysis, and showed dramatic tumor regression in preclinical models. 2-DG completed early-phase clinical trials in glioblastoma and breast cancer. Patients tolerated it well, but hypoglycemia at effective doses limited its usefulness. Neither 2-DG nor 3-bromopyruvate carries FDA approval for cancer. And here is the irony. Medicine already exploits this exact vulnerability every single day. PET scanning uses a glucose analog, much like 2-DG, and tumors gobble it up at rates up to 200 times higher than surrounding tissue. Doctors have been diagnosing cancer through the Warburg effect for decades, and the question now is whether treatment can walk through that same door. That's Switch 1. Switch 2. Metformin and AMPK activation, the diabetes drug that may fight cancer. Metformin ranks among the most widely prescribed drugs on Earth, a staple of type 2 diabetes treatment since 1957. It is generic, costing approximately $4 a month, and a striking amount of research connects it to reduced cancer risk. What metformin does is activate something called AMPK, your cell's built-in energy sensor. When AMPK flips on, it puts the brakes on mTOR, a powerful growth signal we'll cover in Switch 4. On top of that, metformin inhibits a part of the mitochondria called complex 1, lowers circulating insulin and IGF-1, and steers the cell's metabolism away from the kind of fat production that feeds cancer growth. A comprehensive meta-analysis covering 166 studies found that metformin use correlates with decreased risk for gastrointestinal, urologic, and hematologic cancers. Diabetic patients taking metformin show 10 to 40% lower cancer incidence compared to patients on other diabetes medications. And as of late 2023, 57 studies had been registered at clinicaltrials.gov for breast cancer and metformin alone. The caveats matter, though. A large Phase 3 trial, enrolling over 3,600 patients, showed metformin did not improve outcomes in breast cancer patients who didn't have diabetes. And many of the preclinical studies used drug concentrations dozens to thousands of times higher than what doctors can achieve in an actual patient's bloodstream. No FDA approval exists for metformin as a cancer treatment, but the epidemiological signal keeps drawing researchers back, and new trials continue to launch. That's Switch 2. Switch 3. Glutamine metabolism, starving cancer's second fuel. Glucose is cancer's primary fuel, but it is not the only one. After glucose, glutamine is the nutrient many cancer cells consume most, a dependency researchers call glutamine addiction, where at least 50% of non-essential amino acids in cancer cells trace back to glutamine. Cancer cells lean on glutamine for multiple functions at once. It feeds into the TCA cycle to keep energy production running, donates nitrogen for building new DNA and proteins, and helps produce glutathione, an antioxidant that shields cancer cells from damage. Blocking glutamine metabolism amounts to cutting cancer's second fuel line. The lead drug in this space is CB839, also known as telaglenestat, the most clinically advanced glutaminase inhibitor. Trials have tested it in colorectal cancer, triple-negative breast cancer, renal cell carcinoma, melanoma, and AML. Results from using it alone were limited, but combination strategies with other drugs look more encouraging. The challenge is adaptation. When glutamine gets blocked, some cancer cells compensate by manufacturing their own through an enzyme called glutamine synthetase. That metabolic flexibility remains one of the biggest obstacles across the entire field. No glutamine-targeting drug holds FDA approval for cancer. That's Switch 3. We're halfway through, and the next 4 switches include 3, with FDA-approved cancer drugs, and one that may be the most direct reversal of the Warburg effect ever discovered. Switch 4. Pathway inhibition, shutting down cancer's growth master switch. mTOR stands for mechanistic target of rapamycin, and it operates as the central master switch controlling cell growth, proliferation, and metabolism. When cancer hyperactivates mTOR, the consequences cascade. Glycolytic enzyme production ramps up, fat and nucleotide building accelerates, and the cell's self-cleanup system called autophagy gets shut down. The origin story of the first mTOR inhibitor reads like fiction. In 1975, scientists isolated a compound from soil bacteria collected on Easter Island, known locally as Rapa Nui. They named it rapamycin after the island. A molecule from a speck of dirt on a remote Pacific island turned out to target one of cancer's most potent growth signals. Everolimus earned FDA approval in May of 2007 for advanced renal cell carcinoma. Everolimus, marketed as Afinitor, followed in March of 2009 for the same cancer and has since picked up approvals for pancreatic neuroendocrine tumors, certain breast cancers, and other conditions. In its pivotal trial, everolimus cut the risk of cancer progression by 67% compared to placebo, making mTOR inhibitors some of the most clinically validated metabolic cancer drugs that exist. That's Switch 4. Switch 5. HIF2 alpha targeting, blocking cancer's hypoxia survival signal. HIF stands for hypoxia inducible factor, and in clear cell renal cell carcinoma, the most common form of kidney cancer, a gene called VHL goes missing in roughly 90% of tumors. When VHL disappears, a protein called HIF-2 alpha builds up and activates a cascade of genes that drive blood vessel growth, glucose transport, and cell division. Cancer essentially hijacks the body's emergency oxygen response and turns it into a growth engine. The drug that changed the equation is belzutifan, sold as Welireg and developed by Merck. It binds directly to HIF-2 alpha and blocks it from activating downstream targets, earning the distinction of being the first FDA-approved drug to target a transcription factor in solid tumors. August of 2021 brought the first FDA approval, covering VHL disease-associated cancers. The numbers from the pivotal trial tell the story. A 49% response rate in kidney tumors, 63% in brain tumors called hemangioblastomas, and 83% in pancreatic tumors. December 2023 added approval for advanced sporadic clear cell renal cell carcinoma, and May of 2025 brought approval for pheochromocytoma and paraganglioma, making belzutifan the first oral treatment for those tumors. William Kaelin Jr. shared the 2019 Nobel Prize in Physiology or Medicine for discovering the VHL-HIF oxygen sensing pathway that belzutifan targets. Switch 6. IDH mutation targeting, reprogramming cancer's broken enzyme. This switch involves one of the most fascinating classes of cancer mutations. IDH1 and IDH2 are enzymes in the TCA cycle that normally help convert one molecule into another as part of energy production. When they mutate, which happens in about 20% of acute myeloid leukemia cases and roughly 80% of low-grade brain tumors called gliomas, they gain an entirely new and damaging function. Instead of performing their normal job, the mutant enzymes start producing a rogue molecule called 2-hydroxyglutarate, or 2-HG. This oncometabolite jams the cell's ability to read and regulate its own DNA properly, locking cancer cells in an immature, rapidly dividing state. What makes this switch extraordinary is how the drugs work. IDH inhibitors don't kill cancer cells, they reprogram them. By restoring normal enzyme function and reducing 2-HG levels, these drugs allow cancer cells to mature into normal, differentiated cells. Oncologists call it differentiation therapy, and it stands as one of the most elegant concepts in modern cancer treatment. 3 IDH inhibitors now carry FDA approval. Enasidenib, the first IDH2 inhibitor, reached approval in August of 2017 for relapsed AML. Ivosidenib, the first IDH1 inhibitor, followed in July of 2018 for AML, and later expanded to cholangiocarcinoma, a bile duct cancer, and myelodysplastic syndromes. Olutasidenib, a second IDH1 inhibitor, joined the roster in December of 2022 for relapsed AML. Agios Pharmaceuticals developed these drugs, a company co-founded in 2007 by 3 of the most prominent figures in cancer metabolism: Craig Thompson, Lewis Cantley, and Takmak. Cantley deserves a moment here. Back in 1985, he discovered a pathway called PI3K that turned out to be involved in up to 80% of cancers. That discovery earned him the inaugural Breakthrough Prize in Life Sciences in 2013, worth $3 million. The night he made the discovery, he went home and told his family, "This is going to completely revolutionize cancer." And to physicists, this would be like finding a quark no one had ever seen before. And finally, Switch 7. Switch 7. DCA and PDK inhibition, reactivating cancer's mitochondria. We've arrived at Switch 7, the one I asked you to hold in your mind from the beginning. This is the most direct pharmacological reversal of the Warburg effect that exists. Here is how it works. Cancer cells overproduce an enzyme called pyruvate dehydrogenase kinase, or PDK. That enzyme acts like a lock on the door between glycolysis and the mitochondria, shutting down the gatekeeper enzyme called pyruvate dehydrogenase that normally channels fuel into the mitochondria for efficient burning. With that gate locked, glucose gets diverted into fermentation, lactic acid piles up, and the Warburg effect, takes hold. Dichloroacetate, known as DCA, inhibits PDK and unlocks that gate. It forces cancer cells to route their fuel back into the mitochondria, switching them from glycolysis to oxidative phosphorylation. And this is where it gets remarkable. Once the mitochondria come back online, they detect the accumulated DNA damage inside the cancer cell and trigger programmed cell death, a process called apoptosis. The cell destroys itself. A landmark 2007 study by Evangelos Michalakis at the University of Alberta demonstrated that DCA restored mitochondrial function, triggered apoptosis in cancer cells, and shrank tumors in rats. A phase 1 trial in patients with recurrent malignant brain tumors showed the drug was feasible and tolerable, and all patients who completed at least one cycle remained clinically stable. DCA has been in clinical use since 1969 for a completely different condition called lactic acidosis. It is generic, costs pennies per dose, crosses the blood-brain barrier, and has shown the ability to work alongside other cancer drugs like 5-fluorouracil in colorectal cancer and cisplatin in bladder cancer. The reason it isn't approved for cancer comes down to money. No pharmaceutical company has the financial incentive to run the large phase 3 trials required for FDA approval because DCA is generic, with no patent, no exclusivity, and no profit motive behind it. No completed phase 3 cancer trial exists. The main side effect is reversible peripheral neuropathy. DCA doesn't target just one downstream effect of cancer metabolism. It reverses the Warburg effect itself, flipping cancer cells from fermentation back to normal respiration and letting the mitochondria finish the job. Let's bring all 7 together. Switch 1 targets glycolysis directly by blocking the enzymes cancer uses to gorge on glucose. Drugs like 2-DG and 3-bromopyruvate remain experimental, but the principle gets validated daily through PET scans worldwide. Switch 2 uses metformin, a $4 a month diabetes drug, to activate the cell's energy sensor and break growth signaling. Across 166 studies, diabetic patients on metformin showed 10 to 40% lower cancer incidence compared to those on other medications. No cancer approval exists, but the research pipeline stays active. Switch 3 goes after glutamine, cancer's second fuel. CB839 leads the clinical effort, with combinations looking more promising than single-agent use, though no approval has come through. Switch 4 shuts down mTOR, the master growth switch, using drugs like everolimus and temsirolimus, both of which carry FDA approval for multiple cancers. Everolimus cut progression risk by 67% in its pivotal trial. Switch 5 blocks HIF-2 alpha with belzutifan, the first drug approved to target a transcription factor in solid tumors. Response rates reached as high as 83% in certain tumor types across its approved indications. Switch 6 targets IDH mutations with anacedenib, ivosidenib, and olutasidenib, 3 FDA-approved drugs that don't kill cancer cells but reprogram them, forcing immature cancer cells to mature into normal cells through differentiation therapy. And Switch 7 uses DCA to inhibit PDK and reverse the Warburg effect itself, reactivating mitochondria and triggering cancer cell self-destruction. It is generic, inexpensive, and crosses the blood-brain barrier, but it lacks the corporate sponsorship needed for the large-scale trials that would bring it to patients. A lesson runs through all of this that goes beyond any single drug or switch. Otto Warburg spotted something real about cancer in 1923. He had it right that metabolism matters enormously, but he had it wrong that metabolism was the sole cause. Cancer is a shapeshifter. Different tumors, and even different regions within the same tumor, can run on vastly different metabolic profiles. Cancer cells can toggle between glycolysis and oxidative phosphorylation depending on what nutrients surround them, and that metabolic plasticity is the primary reason no single metabolic approach will serve as a silver bullet. That doesn't make these switches useless. It means the future points toward combination, metabolic drugs paired with immunotherapy, precision targeting guided by each tumor's unique metabolic signature, and a deeper grasp of the terrain inside our bodies. The cancer metabolism drug market sat at roughly $18 to $21 million in 2023, projected to climb past $8 billion by 2032. This field is accelerating, not winding down. Ralph Dibber-Ardinis and Karen Vousden, two of the leading voices in the field, wrote in 2024, with modern tools and an incredible amount of talent focusing on these problems. The upcoming decades should bring transformative discoveries, and who knows how Otto Warburg would react to learning that his work on glycolysis and respiration launched an entire field of cancer biology, still accelerating after a century of discoveries. 100 years ago, a difficult, brilliant, horse-riding German scientist peered at cancer cells through a manometer and noticed something that didn't add up. The cells were devouring sugar, as though they were starving, even when they weren't. 70 years passed before the medical world took him seriously, and now, a full century later, his observation underpins millions of cancer diagnoses every year, has produced multiple FDA-approved drugs, and has opened 7 distinct pathways for slowing cancer's growth. Warburg put it in his own words during a 1966 lecture to fellow Nobel laureates. Cancer, above all other diseases, has countless secondary causes. But even for cancer, there is only one prime cause. And the prime cause of cancer is the replacement of the respiration of oxygen in normal body cells by a fermentation of sugar. He overstated the case. But the core insight, that cancer's metabolism is different, and that difference can be exploited, may stand as one of the most consequential observations in the history of medicine. If this video helped you understand something new about how cancer works, share it with someone you care about, and keep in mind that these are research developments, not replacements for medical care. Talk to your doctor, stay informed, and never stop asking questions.